BLOSUM. Okay. The BLOSUM matrix, the concept of a block substitution matrix. It's a tool in bioinformatics. You want to score the evolutionary divergence of proteins based on alignment of many sequences. Okay? You're trying to figure out how different these things are. Okay? You're trying to also, what we want to do in our class, we want to see how similar two or more proteins actually are. We want to figure out the frequency with which a single amino acid would substitute for another one in nature. Okay? So glutaval wasn't ideal. This was our sickle cell anemia example. Glutamate had a charge on it. Valine is hydrophobic. This is something that probably isn't going to be sustained and supported in nature very frequently, but a block substitution matrix would help us identify that. So here we have myoglobin, hemoglobin alpha chain, and hemoglobin beta chain. Okay, and they all had sort of similar tertiary structure. But their actual sequence, protein sequence, primary sequence, looked very different. Only the green represented absolutely conserved or perfectly conserved across all three amino acids. Same position, same amino acid. All the white spots represent some type of change, and the dots represent a gap in the alignment simply because these aren't perfectly the same length or number of amino acids. If you see the word sequence identity, if you'll be asking for sequence identity, this is the equation you're using. Number of identical amino acids, same position, same amino acid, over the total number of residues in the string. Now for your homework, this is pretty easy. There were only 6 to count up. They all had 6. There wasn't any of this misalignment. And again, you'd be given something a little bit simplified in that way. And so then you could say that these are, for this one, I didn't actually count them, but 20 to 30% identical. So you can look at more. Okay? So despite near perfect tertiary structural conservation, right? Tertiary structure, the overall fold, looks pretty similar, but the underlying sequences are very different. And so here was all of our top 3 chains that we just looked at, but then we have all of these other species involved too. We're looking at a longer sequence of chains. Only 2 positions were absolutely conserved, 100% conserved. You see those words, that's what I mean, perfectly, absolute, 100% all the way down that position. Every single thing in the block is the same, okay? And then the other green to yellow is indicating like fewer changes. Okay? And so when you get a lot of sequence together of a sufficient length, you can start to recognize patterns of things that frequently substitute for each other. Stuff that can substitute and still fold up the way you want it to and still function the way it needs to. So BLOSUM, we're looking at a quantitative measure of similarities. Okay, so the scores in the matrix are SIJ, substitution score. It is a score relating a replacement of the ith amino acid with the jth amino acid. A positive score is a quantitative measurement of similarity. Negative scores represent differences. Okay? So the substitution of one with another that has similar properties probably means it won't have an effect on protein folding. So that's why we want to look at these things and see how over time, even though something substituted a lot for something else, we're still getting folding that we expect, we're still getting the function we expect. So when you look at these scores, keep in mind that an identical amino acid will always have a higher number than any substitution. And a conservative substitution, so something with similar properties, will always have a higher score than a non-conservative substitution. Okay? 2 things that have very different properties. Okay. So again, there's only a few amino acids that have to be absolute or 100% conserved, just 2 positions across all of those globins, because they interact with the heme group. So the most important stuff tends to be conserved for function. Everything else sometimes can be swapped out, and it'll still be able to fold the way you expect. Okay. So if you see conservative substitution, this is the replacement of an amino acid with one with similar biochemical properties. Non-conservative is we're talking about maybe something that's large or charged, being replaced by something small or neutral, or vice versa. And that's what we expect to see. Is this happen way more frequently in nature on our scoring matrix than this? We want to ask what substitutions are tolerable and how often do they happen. To get good statistics on these probabilities, we have to have a lot of sequences, thousands of sequences of sufficient length. We don't want them to be too similar. So this is where 62% comes from. Sequences that are greater than 62% identical will throw out, okay? Because otherwise it's going to skew the results. We're going to think maybe that things are substituting more frequently than they really are. Okay? 62 is an arbitrary cutoff. It's based on people looked at the matrices, looked at the math, and were like, okay, this is a number that we're happy with, and it's just a common choice. A couple other options. You can make a BLOSUM 80 matrix. You'd have closer related proteins. You could make a BLOSUM 45. You could look at stuff that's even further apart. You can make whatever you want. Like I said, 62 is just a popular choice, and it's the only one we're looking at. Okay? Entries in the matrix are called the substitution score, or SIJ. They relate that frequency with which the one-to-one swap happens. The substitution scores are related through this equation here to something called a likelihood ratio, and sort of the qualitative interpretation of the likelihood ratio, because I won't make you calculate it, is the frequency of an I to J substitution in the alignment of those related proteins over the frequency with which those two would substitute if you scrambled everything up and let it happen randomly. Okay? And so if the frequency with which it actually happens in your aligned sequences, and if you randomized absolutely everything, is the same, 1-to-1, then what you're saying is that substitution is about as likely as random chance. And L of 1 means that change is just as good as random chance. And if you take the log of 1 here, you're going to get a 0. So a 0 score in here means A, alanine substitutes for cytosine, just as good randomly. Positive scores mean it happens more frequently. Negative scores means it happens less frequently. And this was the example that we saw from last time. Again, the reason we would use this matrix, what do we do with this? Figure out if two things are closely related. Are protein 1 and 2 more closely related than 1 and 3? And we can go into the scoring matrix, look at, for example, a change from alanine to methionine. That had a score of -1. Look up the next pair. Sum them all up. Do the same thing for our other pair. Okay? And I think everyone turned in the homework, so you should have done this across your worm to zebrafish, worm to human, whatever it was. Okay? Then plug into our likelihood ratio. Rearrange that equation. Proteins 1 and 2 are more closely related by about 45 times. And again, why do we care about any of this? I think I'll just have a question that's like, other than water, the other reason we do any of this is to predict or model protein structure. So if you have a sequence of a protein of interest that you don't necessarily know its structure, but you can align it to other proteins, and you're seeing that you have percent identical residues over a sufficient length, you're in the region of, hey, I bet it has the same structure as that protein. Yes?
How can you have the region on the top left where 100% of the residues are identical, but you can't tell if they would have the same structure when they have to?
For a really short chunk, think about random chance. Like the likelihood, the odds of having, 5 sequences in a row that perfectly match. 5 sequences also don't equate to any overall structure, really. It's sort of, there isn't enough structure there yet. So you can't say enough with only 5. You need like the statistical power of more length.